Okay, so we know what our lymphatic capillaries, lymphatic vessels, our thoracic duct, our right lymphatic duct are, what our cisterna chyli is, what lymph nodes are, and that tonsils are specialized lymph nodes. We have three types: the pharyngeal, which is what you'd call your adenoids, the palatine, which is what we typically think of as being our tonsils, and then the lingual, which lingual means tongue, so they're at the base of the tongue. So what we're going to look at now are the last four groups of words here: the spleen, the thymus gland, the mucosa-associated lymphoid tissue, and particularly the Peyer's patches. Okay, so here's our spleen, which is found on our left side of our trunk. So the spleen is actually made up of reticular tissue and made up of these nodules, and has what's called red pulp and white pulp. Now the red pulp is basically where the blood is being filtered through the spleen, and of course one of the major things we're doing in the red pulp is we're removing the old blood cells that have been circulating, the red blood cells that have been circulating for around 120 days, become damaged. So we tend to filter them through the red pulp and remove and recycle parts of the old red blood cells. But the white pulp is actually where we tend to have our white blood cells hang out, very similar to what they did in our lymph nodes. So just like our lymph nodes is basically filtering and cleaning our lymph, that fluid that's derived from our blood, well then our spleen is basically filtering our blood and also having an area that our white blood cells can hang out inside of our spleen, so that if we do have infections, the infections, as they circulate through the blood, will circulate through the spleen, then we will activate the white blood cells that will be in the white pulp areas of that. So again, without going into too much detail, the spleen is very important for immunity. It also is an extra reservoir of blood. If we're ever in an emergency, we need more blood to our muscles. We have quite a bit of blood that is here that to be able to be used. You can live without your spleen. People have splenectomies. An infection that's very common in the spleen is mono or Epstein-Barr virus, which basically causes the reticular tissue to kind of spread out, kind of like a sponge, and our spleen becomes engorged in blood, and because of the effect of the virus on the tissues. And then the thing that can be dangerous is if something were to injure the spleen while it's engorged with blood, then you can have a major blood loss. Now, at the same time, you're going to be very tired if you have mono or something, because you have so much blood that is in the spleen at the point that you have mono, that you have less blood for the rest of your body. And anytime you're anemic or have less blood somewhere, you're not going to have the same energy levels that you would when you have blood distributed the way it's supposed to be distributed through the body. Okay, so the spleen, again, is filtering the blood in the same way the lymph nodes filtered the lymph. And it's another source of an area for white blood cells to hang out. So as the infections or the antigen-presenting cells activate the white blood cells, especially in the white pulp areas, then that gives us an extra added immunity source to help take care of infections in the body. Again, one of the other major jobs of the spleen is to replace the old red blood cells, which only have about a 120-day life cycle. Okay, another immune organ is the thymus, not to be confused with the thyroid gland. It does sit kind of in the top part of the heart, kind of embedded in the pericardial areas. And what the thymus is, it is a gland where our T cells that have been originated in our bone marrow, they migrate to the thymus, where they are actually differentiated into T cells. Now that's kind of a long, quite complicated process, but our T cells are actually called T cells because they are thymus. Now remember, T cells and B cells are actually blood cells called lymphocytes, and they do what's called specific immunity, which is a very important job. And if we can't do this job, we're not going to be able to fight off infections. So one of our T cells is called a helper T cell. Well, that happens to be the cell that HIV affects, and so by affecting this controlling cell called a helper T cell, then it also affects both the B cells and the cytotoxic T cells, keeps them from doing our job. And as we know, full-blown AIDS is basically a death sentence. And so just trying to throw that in there to show you that T cells is where very important cells, or the thymus is where very important cells called T cells develop and are basically educated and become T cells, and then leave here going through the bloodstream where they then have the ability to fight specific infections by killing infected cells. So the thymus is very important for development of your T cells is the bottom line. Okay, MALT, also known as mucosa-associated lymphoid tissues. MALT is what lines your internal structures, especially your intestines. So we know that a mucosa, a mucous membrane, lines every entrance and exit to the body. Well, since it lines every entrance and exit to the body, that means that it's easier for things to enter the body through the lumen of the mucosa. Therefore, we have lymphoid tissues that are actually inside of these mucosal areas that help fight anything that gets into our lumen, our entrances to our body. So our MALT is basically what is protecting us, especially through our digestive tract. Now we have a specific area in the intestines in particular, and more particular within the ileum of the small intestine that is lined with lymphatic tissues that's actually called Peyer's patches. And what that primarily does, that primarily keeps the E. coli bacteria, which is actually in our large intestine, from backing up into our small intestine. So that's a subject we'll talk more about once we get into the digestive system. Okay, so make sure you're familiar with this terminology and where it's at on the pictures. Again, look at the structure list, see if you can identify where those things, what those things are and where they're at.